Good day. This video abstract is for the review article entitled Involvement of DNA Methylation in Tissue Regeneration upon Liver Injury, which I am presenting in behalf of my co-authors, Dr. Claudio Tiribelli and Dr. Cecilia Sukawati, under the institutions Fondazione Italiana Fegato and the University of Trieste. In liver regeneration, very distinct patterns of gene expression are required to trigger the activation of a quiescent liver to a regenerative one. Following injuries, such as your partial hepatectomy, hepatocytes turn on a pro-regenerative switch to undergo synchronized cell proliferation and restore liver mass. Since epigenetics takes part in the maintenance and control of gene expression, its function in liver regeneration is also pivotal. This review will briefly describe the significance of the DNA methylation regulators, DNMT1 and UHRF1 in liver regeneration following liver injury. UHRF1 acts as a multifunctional epigenetic regulator, allowing the crosstalk between DNA methylation and histone code. UHRF1 is required for DNA MT1 localization, functional activity, specificity, and stability. UHRF1 has been identified as a regulator of cell proliferation in several tissues and is equally important as a regulator of hepatocyte proliferation. DNMT1's ability to maintain the methylation patterns in the daughter cells during replication is heavily dependent on the functional binding and recognition of UHRF1 in a hemimethylated DNA. In a normal quiescent liver, hepatocytes do not undergo cell cycle and pro-regenerative genes are repressed while differentiation genes are active. Upon liver injury, a proliferation switch is turned on in hepatocytes to compensate for the loss of tissue. This dictates the cells to enter the cell cycle phase where DNA replication happens, and the maintenance mechanism of both UHRF1 and DNMT1 is necessary to ensure the proper methylation pattern throughout the regenerative process. Since the roles of both the UHRF1 and DNMT1 have been identified in terms of proliferation and maintenance and as markers of stress response, this could be meaningful information that can help utilize in the utilization of these mechanisms in the clinical setting.